Hello everyone, welcome to our radiochemistry and PET imaging showcase highlighting our capabilities here at MBI and ARA-MBI. My name is Robert and I'm the facility manager here at ARA-MBI. The program that we have for today is I'm first going to give you a quick overview of our facilities, our capabilities and how you go about accessing those. And then what I'll do is I'll pass on to our other two speakers, Dr. Maggie Allsbrook and Dr. Bianca Jupp. They'll go through in a bit more detail about our capabilities and how you can apply those to your research. So just to give a quick overview of who we are as MBI and ARA-MBI, we've got two site locations. One is located at the Clayton University main campus, and the other is located at the Alfred Hospital as part of the Alfred Research Alliance precinct. We have two Medizo nanoscan systems, and they're both equipped with single and multiple rodent beds. That way we can do multiple animals at a time. And we've also got a recent upgrade for a marmoset bed that allows for marmoset imaging. In terms of other support equipment, aside from these main scanners, we have a Twilite arterial blood sampling system. We've got an autoradiography scanner that has a dedicated hot cryostat on site as well. And we've also got to minimise your exposure to radiation when you're using these counters. In terms of expertise, we've got a range of PET-CT scientists available, along with radiochemists. So we can provide that expertise from the very early projects of the early stages of the project, and things like developing your radiopharmaceuticals, but then also with the application side of things. And very closely related to that, in terms of the networks we're part of the National Imaging Facility and also the Victorian Biomedical Imaging Capability. And we've recently launched a new network known as the Melthera Research Lab, which is a collaboration between Monash University and the Helmholtz Institute in Dresden. And lastly, we do also have a translational research focus. So for us to be able to translate that research, we've got access to an in-house Siemens Biograph PET MR system. And the Twilite system is also compatible with clinical studies. So how do you go about accessing our facilities? Well, I'll give you a quick overview of how we do that. So we're a fee-for-use service, and access is available to all other universities and research institutes within Australia, and also available for industry and commercial users as well. We can provide assistance with ethics approvals. We can also provide small-scale pilot studies or proof of concept studies at reduced rates. You know, if it's a new target that you're trying to track or something like that, we can make those accommodations. We can also provide training to users so they can use the facility unsupervised. Periodically, we have grant schemes that run to help new users come into the facility and start up some projects. So those are things that we advertise through our LinkedIn, Twitter, our email lists. So keep an eye out for those. And then if you've got any other information that you want to know, or if you've got any other questions, you can reach out to our main email accounts, which are displayed on the screen now. So what I'll do next is I'll pass on to Dr. Maggie Allsbrook, who will give an overview of our facilities at our Clayton site. Maggie.
Thank you, Robert, for the wonderful introduction to our facilities and capabilities. It's really great to see such a diverse audience online, so thank you for tuning in. Good afternoon, everyone. I'm Maggie Allsbrook, and I'm the Senior Radiochemist at Monash Biomedical Imaging, leading the radiochemistry capability at MBI. I had some sharing difficulties, so I'll be requesting Robert to just click onto the next slide, and thank you for your patience with that. I'm going to begin my half of this webinar by introducing the radiochemistry facilities that we have currently at MBI. We were incredibly lucky that our facilities were recently expanded upon with the establishment of a manual radiochemistry suite shown in the left-hand picture here. This laboratory was officially unveiled earlier in the year, a part of the Helmholtz and Monash International Partnership that Robert mentioned earlier. It features a fully lead-lined fume cupboard and a range of radioanalytical instruments. We also have a hot cell laboratory for limited manual and automated development work. This hot cell laboratory also has its own adjoining radioanalytical laboratory with a range of instruments as well. We lastly have a radiopharmacy dispensary. This area is utilized by the clinical and preclinical teams to draw doses for human and animal studies. And if you wouldn't mind going to the next slide, Robert, thank you. One of the main functions of the radiochemistry platform is to support MBI's imaging facilities. On site at MBI, we have a Medizo PET-CT nanoscan for preclinical imaging. We also have a Siemens PET-MR biograph, which is used for human imaging and large animal studies. The radiochemistry team and the preclinical team work very closely with one another, and our facilities are co-located. It's this synergy between the two teams that is one of the major advantages of working with MBI for preclinical imaging studies because of the close relationship between tracer and imaging. And if you wouldn't mind going to the next slide again, Robert. We have an array of on-site radiochemistry equipment, the most exciting, of which are highlighted on this slide. We are very fortunate to have our own gallium-68 generator on site at MBI to facilitate studies with this radionuclide, which is currently the most frequent request we receive from users. We have multiple dose calibrators, radio TLC and radio HPLC instruments from Shimadzu and LabLogic, which facilitate radiopharmaceutical development and quality control. We also have multiple gamma counters at MBI, including the newest HiDex automated gamma counter. This instrument automatically measures the gamma radiation emitted by a radionuclide and weighs samples. This instrument is incredibly powerful in facilitating a range of assays, including fast and effective biodistribution studies. So we are very lucky to have one of these on site. And if you could switch over to the next slide. With all the instruments we have at MBI, we are able to perform the measurements summarized on this slide, all of which can be developed and performed in-house self-sufficiently. We are able to perform and develop a range of quality control procedures. We can report radionuclidic identity and purity, radiochemical identity and purity, radiochemical yield, chemical purity, specific and molar activity, pH, and we can facilitate gas chromatography measurements if it is of interest to users who are developing a radiosynthesis for clinical application. Being in such close proximity to the preclinical team, we are able to perform an array of assays. We can, of course, perform imaging studies for small and large animals, as mentioned earlier. We can perform biodistribution studies, radiometabolite stability and immunoreactivity assays. We also have the capability of performing theranostic studies. If you could switch to the next slide, please, Robert. This field is highly regulated, so I've included this slide so you are aware of the radioisotopes that we can work with. We are actually in the process of updating our radiation license and are very close to submitting our update. So this list is indicative of the isotopes that that will be included in the update. We can handle and facilitate work with carbon-11, fluorine-18, scandium-47, copper-64, gallium-68, copper-67, gallium-67, zirconium-89, technetium-99M, iodine-123, 124, and 125, terbium-161, thallium-201, and lutetium-177. These radioisotopes have a range of applications in imaging and therapy. If you are interested in knowing more about these radioisotopes and their potential applications and properties and how that might complement your research, please do feel free to get in touch with us. We are not limited to work with radioisotopes. We do regularly work with radiopharmaceuticals and traces for preclinical and clinical studies. These can be manufactured in-house or ordered from external sources. Radiopharmaceuticals and traces that we regularly access at MBI are FDG, lutetium-177 PSMA, FPSMA, copper-64 ATSM, and a range of technetium-99M traces. Please keep in mind that we are user-driven. So if you would like to work with a radioisotope which is not featured on this list, we can always expand our license in order to serve your research. But the license update does take some time, so please be proactive. It's essential just so we can meet your timelines of your research. And I'm ready for the next slide. The Radiochemistry Platform at MBI can provide the following services to users, and we obtain requests from a range of backgrounds, including fundamental researchers, commercial organizations, from industry and clinicians. We can provide contract radiochemistry development services. Platform staff have significant experience in the development of traces for clinical and preclinical application. We can deliver those drug candidates developed to preclinical imaging and therapy studies. We can support clinical translation, including stability and formulation studies, process design, translation onto a synthesis unit, and scale-up support. We also provide access to nuclear facilities where users can perform their own studies, and they can receive training from our staff if required. We also provide consultation services, providing expert advice on a range of aspects such as radiopharmaceutical development, GMP compliant manufacture, cyclotron facility operations, design and workflows, to name a few. And if you want to switch over to the next slide, please. The main request the platform currently receives is around product development. So I'd like to go into more depth on this process and what the platform can offer to you as a user. Platform staff have extensive experience in the development of small molecules, proteins, peptides, and antibodies, having successfully taken each class of radiopharmaceutical from the initial labeling development to authority approval to manufacture for clinical trials. And I've summarized briefly this development process on the right-hand side of this slide to give you more of an idea of what is involved in this process. So generally, when developing a product, I would start by developing the radiolabeling and purification process. Subsequent stability and formulation studies would then be performed. Manual production is possible, but typically for clinical production, it would be translated onto a synthesizer, which includes writing the sequence. I've developed the QC protocols and release criteria. We then work with the investigators on the approval for the study to proceed, including generation of SOPs, validation runs, and submissions to regulatory bodies. And once that approval has been granted, we then proceed to clinical production runs. The Radiochemistry Platform at MBI can work with users to deliver radiopharmaceutical products and provide advice on any aspect along this workflow. And the pipeline is more broadly summarized on this slide. Currently, the MBI Radiochemistry Platform can deliver to every aspect along this pipeline. And if you just want to go to the next slide, Robert, there is one major exclusion along this workflow, and it's the fact that MBI cannot currently manufacture a radiopharmaceutical for a clinical trial. However, MBI is very excitingly closing this gap within its capabilities, and I'd like to spend a few minutes just outlining this future outlook. So if you would go to the next slide, please, Robert. MBI is in the process of establishing the Australian Precision Radiopharmaceutical Facility, or the APRF. This is a brand new radiopharmaceutical development and manufacturing facility and will be an expansion of the Monash University Innovation Precinct. It will be located at the MBI site and is strategically located in close proximity to the Victorian Health Innovation Centre and the New Heart Hospital. This facility will establish itself as a leader in the manufacture of radiopharmaceuticals for research and clinical application. It is anticipated to be delivering to research and development work by the end of 2026 and manufacturing GMP-compliant radiopharmaceuticals by early 2027. The APRF will house an 18 to 24 MeV cyclotron and will produce a range of PET radionuclides in-house. We will manufacture the radionuclides shown on this slide: carbon-11, nitrogen-13, fluorine-18, copper-64, gallium-68, and zirconium-89. That will be produced off our medical cyclotron. I do want to stress that we will not be limited to working with just those radionuclides and, just the radionuclides that are produced off our cyclotron. We will be able to work with a range of externally manufactured radioisotopes, including therapeutics. The planned facility will be 1,500 meters squared in gross floor area, which is about the size of 6 tennis courts. So it is a substantial size. The facility as a whole will have 3 dispensing hot cells and 7 double stack synthesis hot cells to facilitate the manufacture of radioisotopes and radiopharmaceuticals. Again, that's just to give you an idea of the size of the facility. We will also have other key pieces of equipment, including shielded fume cupboards and laminar flow hoods, as well as multiple radioanalytical suites. And if you could just click over to my last slide, please. The APRF will focus on the delivery of 2 main service capabilities. These are the expansion of the radiochemistry services provided through the delivery of dedicated radiochemistry research and development facilities. The APRF will also deliver GMP-compliant production facilities for the delivery of GMP-grade radiopharmaceuticals to early and late-stage clinical trials within Australia. Together, these aspects of the facility will provide crucial infrastructure to allow Australia to become world leaders in the development and clinical evaluation of novel radiopharmaceuticals. And then that's the end of my slides, so I'm going to hand over to Bianca.
Thank you so much, Maggie, and thank you, Rob, and thank you all for joining us today. For those of you who don't know me, I'm Dr. Bianca Jupp. I am the lead PET-CT scientist here at ARA-MBI facility, and I'm involved in helping you as a user develop and optimize your PET study, including everything from training in the specific animal facilities and in radiation handling, and in designing your optimal analysis strategies, facilitating access to specific radioisotopes, but basically here to hold your hand through the wonderful world of PET. So today I thought I'd take advantage of your time today to give a bit of a PET 101. And so hopefully you can walk away at the end of this with a better understanding of what PET can potentially do for your research. Okay, so by way of a bit of a preamble, why do preclinical imaging in the first place, besides the fact that we want to make pretty pictures that will hopefully end up on the front cover of very high impact journals? I think the biggest power that we have, in terms of preclinical imaging is this idea that it's directly translatable. that we can, the approaches and techniques that we use preclinically are essentially identical to what we apply in the clinic, giving us the capacity to forward translate our findings from our preclinical models to the human condition. But the flip side is also possible. We can translate back again. So this is really important because we can take what we see in the clinic and see whether or not it also is apparent in our animal models. And this gives us the capacity to validate, providing face, construct, and predictive validity for our animal models, and also giving the capacity for us to improve, to make them more clinically relevant, hopefully resulting in better outcomes for these studies. The other major advantage of preclinical imaging is the idea that it's non-invasive. So first and foremost, this gives us the capacity to perform longitudinal studies, so we can look at how things change over time. So we can gain insight into potential etiological or risk mechanisms that underlie risk for specific diseases. We can identify biological markers of treatment response or treatment prediction. We can map disease ontogenesis, and finally, we can assay treatment response. Given the fact that imaging involves the three-dimensional, large, in many cases large field of view applications, we can start to ask potentially regionally non-hypothesis-driven questions. As I said, these are large field of view, and in many cases within our animals, we get visualization of the whole body, giving us the capacity to look at regions that we wouldn't necessarily be able to, or that using at, say, a histological approach. So we can look at whole body, whole organ, and then indeed multi-organ systems, and giving us the capacity to start to apply a systems biology approach to our questions. And finally, because we are working with preclinical models, we have the capacity to actually correlate these imaging findings with our post-mortem measures, providing greater information than we would to our potential pathological mechanisms of our disease state. So there is a whole host of imaging modalities available to us within MBI and ARA-MBI, but why PET? And we often have this debate with my colleagues about why PET is so great. It doesn't just make pretty pictures. In fact, it has an unrivaled capability to sensitively and specifically visualize and measure molecular targets and physiological processes in the body. And it does this in a quantitative manner, both on the macro and micro scale. And this is just an example here. This is FDG PET and the corresponding structural MRI. It's the same individual. And while there's really no indication of any structural changes, what we can see here quite distinctly is a reduced uptake of this radiotracer, and this actually happens to be an example of Alzheimer's disease. So why is the sensing function important? Obviously, I've given you an example for Alzheimer's disease, particularly given the fact that, you know, it's a classic of biology that form follows function. So surely changes in structure must relate to two changes in function. But we often know, and this is a wonderful example, given as an example here as the Homer car, that this is often the exception rather than the rule. So, for example, pathological states we know can occur without visually apparent structural alterations. And when I say visually apparent, yes, okay, maybe down on the microscopic level, but as far as imaging is concerned, we don't necessarily see this. For example, in the case of neuropsychiatric disorders. As I showed you before, functional change can actually precede structural change. And so by assessing function, we can look early in a disease process that we wouldn't necessarily be able to see using structure alone. And also by understanding function and by affect dysfunction, we have the potential to identify potential therapeutic targets for the development of treatment approaches. It also gives us the capacity to understand therapeutic mechanisms of action and monitor treatment response. So hopefully I've convinced you now that PET is the power, is a wonderful approach, and I will put my hand up and say I am a neuroscientist by trade, so a lot of the examples I'm going to be using today will be very neuroscience heavy, but PET has broad applicability across a number of medical sciences, including cardiology, more classically oncology, for example, monitoring treatment response, tumor treatment response. There's an emerging field in immunology and orthopedics, and by far I think the largest bread and butter for the use of PET has come in the pharmaceutical imaging industry, where it has been used to great effect in drug development for monitoring drug pharmacokinetics, biodistribution, and pharmacodynamics. So what is PET? I've been rabbiting on about this for a while. Basically, it stands for positron, which is a form of antimatter. It's an anti-electron that is emitted, i.e. emission, from an unstable radioactive material, and tomography, which is basically Greek for slice right. So we collect a number of different slices through our body using this positron emission, so PET. Going to go to the next slide. There we go. So how does PET work? Essentially, we start with these very specialized molecules known as radiotracers. And these can come in a variety of flavors. As Maggie introduced earlier, we have things such as small molecules and antibodies, single-chain peptides. But essentially, what they do is that their common denominator is that they've been labeled with a positron-emitting isotope. Once our very fantastic chemistry teams have created these radiotracers, they're delivered to us in the PET facility where they're administered to our subject. The radiotracer then circulates through the body, and what the amazing power of PET is the fact that these radiotracers accumulate at its specific molecular target. And so this really gives the possibility for PET to label any molecular target within the body, which at the moment is limited to how good your radiochemistry team is. Which, with Maggie on board, we are going great leaps and bounds to be able to really provide traces for your specific application. But yes, okay, your tracer is accumulating at your molecular target. What happens next? So we can see here our little rat is in the PET scan. And remember that these radioisotopes are undergoing this constant decay. And what this relates is that it causes this release of these positrons, which then meet up with their matter counterparts, electrons, at which point they destroy each other. There's an annihilation event. And what happens is you get the emission of two gamma rays that are pretty much 180 degrees from one another. And that when this occurs within the PET scanner, using some very fancy detectors that are made of crystals and some great electronics and lots of maths, we can actually get a readout of where in time and space these annihilation events are occurring, which ultimately ends up being a three-dimensional map of how much radiotracer was in a specific area for the duration of the PET scan. And this is what the output looks like. Okay, so what information can I get from PET? So first and foremost is this idea of target availability. But this involves, there are many things that actually build up to this idea of target availability. First of all, quantity, how much target is there? And so this is an example of an amyloid scan on the right, and we can see it's just simply trying to look at, is there amyloid there? And we can see that in the bottom scan compared to the top scan. But beyond this, there's also the issue of function. So how well can the target bind or be taken, take up the radiotracer? And so this is another example here of the radiotracer flumazenol, showing that differences or changes in receptor, what am I trying to say here? The receptor subtype actually can affect how well that tracer binds. And so by that way, we can get a readout of how well that receptor is working. There's also the role or the influence of endogenous ligand and substrate to that target availability, because we know that endogenous substrate can compete with the radioligand at the target. And so we see an example here of this is FDG PET, and on the left we have the same individual, but he's got a high blood glucose, and we can see that actually influences how much of the radiotracer is taken up in the brain. So if we compare it to the right, the same individual in a hypoglycemic state. And based upon this capacity of endogenous ligand and substrate to compete with radioligand, we also have the capacity to test how much endogenous ligand may be released under, say, a pharmacological or a physiological stimulus. And the example that I've shown here is the displacement of a radiotracer for dopamine D2 receptors in the striatum following the injection of amphetamine. So we know that amphetamine increases dopamine release within the striatum, and we're able to actually show it here using PET. So the endogenous dopamine displaces the binding of the radiotracer. So lots of exciting applications. We can also do drug biodistribution, pharmacokinetics, and pharmacodynamics. And as I mentioned earlier, this is usually the approach that's taken for drug development. So we can look at where my drug goes after I administered it. So in the image on the bottom here, there's a very cute study where we've actually labelled polystyrene with a copper-64 isotope and fed it to these mice to see where it goes and how long it stays. Finally, we can also look at the pharmacodynamics, and so that's looking at how well my drug interacts with its specific target. And so in the example I've got here, this is a novel drug that's being used to try and model the effects of the opiate buprenorphine. And we can see that with increasing dose range, it displaces the binding of that radioisotope. So showing that the drug that they've developed is very specific for the same target as buprenorphine. In the same way, we can actually identify the best dose of drugs to give based upon what we know should be target occupancy for a receptor of interest. Okay, so as I said, PET basically relies, is dependent on the capacity to produce these positron-emitting isotopes. Now, these are exceptionally, or have a tendency to be exceptionally short-lived, which really limits the potential to utilise them within the lab. Now, obviously, this is not going to become such an issue once the cyclotron is up and running at MBI, but for now, both at MBI and ARA-MBI, we are limited to the use of radioisotopes that have been labelled with specific positron emitters. And I've just highlighted the ones here that we generally get access to and use in the lab. So once our radioisotope has been developed in or produced in a cyclotron, it then gets shipped for radiochemistry and formulation. And Maggie very nicely covered the facilities that we have on offer or will have an offer at Clayton in the very near future. But the biggest things that we on the PET side of things have to worry about in terms of our radiochemistry and formulation is first, how much are we going to get? We are described by law that we can only handle so much radiation in the facility at any one time, but it also builds into how many scans you can potentially do in a day. And this also builds into the idea of specific or molar activity. And this is a measure of how much radioactivity exists per mass of compound. And this is really important because in order to, well, the way that we use PET, essentially our radio tracers are just that. They're drugs that are provided in tracer quantities, so usually nanomolar range and below, usually around femtomolar range. And this limits the potential off-target pharmacological effects of these drugs, as well as potentially issues to do with toxicity and side effects. So, what PET tracers are available to users? So first and foremost, we are lucky enough here in Victoria to have access to a number of commercially supplied radio tracers through the company Cyclotech, which is located in Bundoora. And we in the facility have the capacity to be able to put a order through Cyclotech to provide these radio tracers. So I'm just going to flick through a couple here quickly. However, you may think that that's perhaps a little bit limited, and there are a large array of radio tracers that have been developed and validated that we can potentially use on the basis of their using a fluorinated isotope within the facility. Some PET tracers for oncology. But of course, the problem is that in doing so, we don't, well, until our facility is up and running, we don't necessarily have access to these tracers, so we need to go out and talk to our wonderful radiochemistry labs within Victoria to give us a hand. So to start, I'll give you an overview of what we currently have in use at ARA-MBI. So firstly, we are doing FDG for glucose utilisation, which is provided by Cyclotech. We have a number of collaborative tracers, so FBR for neuroinflammation, altancerin for serotonin 2A receptors, clomazanil for GABA-A receptors, ATSM, which we believe is a marker for oxidative stress, and then some single-chain peptide / antibody work that can be potentially catered or developed to your specific amino acid sequence of interest. Also in development, we have SMBT1 for astrocyte activation, FDEX for muscarinic receptors, some copper-64 labelled oligonucleotides for biodistribution, and perhaps something for your favourite target. And of course here I have to acknowledge the absolutely fantastic radiochemistry collaborators that I've had the pleasure of working with, and hopefully we can help facilitate something excuse me, for your particular needs. So in considering radiochemistry for future users, the first thing that I think you probably need to consider is it available commercially? Can we go to Cyclotech and tap them on the shoulder and say, you know, can we get this off the shelf? And if it isn't, the next point is to go to the literature. Has it been described? Do we know its synthetic route? And this will make life much easier than trying to develop something from scratch. But of course, it's going to require collaborative agreement with our radiochemistry groups. And this comes to logistics. First and foremost is the idea of production schedules. So, for example, copper-64 isotope is only available on Tuesday and Wednesdays. There's limited time of our wonderful radiochemistry teams to be able to produce radiotracer for you. And so realistically, even if using a commercial supply, you might only be able to get access to your radiotracer once, maybe twice a month. So worth kind of taking that to heed in designing your experiments. There's also issues, as I alluded to before, in terms of radiochemical yield and molar-specific activities, which can limit how many scans you can get through for a particular production. And then, of course, we don't want to talk about it, but it is a common feature of PET is that the radiochemistry is expensive. And then finally, we also have to consider the analytical method development and validation, which also plays into how long your scan times may be. So, turning to analysis and validation, how do I turn my pretty PET pictures into data, into numbers that I can, do p-values on and publish in my high-impact publications? And so while it's not necessarily so much of an issue for our antibody-based radiotracers, which tend to be very slowly metabolized, have slow kinetics, and as a result, simply quantifying what we would call a standardized uptake value in many cases at a single time point, in many cases, is sufficient. But for our small molecule radiotracers, so things that are more likely to look at receptors or enzymes within the body, what the PET camera sees is actually heterogeneous. So it's not just the amount of radiotracer that's specifically bound to the target. In fact, it comes in a variety of different measures. So we have activity that is specifically bound to the target, that is free and floating around in the tissue, and that is also non-specifically bound in the target. And often there's a small component that is also the radiotracer that's within the blood. So this is problematic because it brings out a potential bias in our measurement. We're not actually measuring what is bound in the tissue. It's a combination of all the above. And this can really limit our accuracy. And so this is where kinetic modelling potentially comes to the rescue to stop your PET study from ending up in the PET cemetery. So kinetic modelling involves a dynamic scan. So instead of administering our radiotracer, waiting for it to do its thing, and then scanning our animal, we administer and scan administer the radiotracer and scan immediately for the duration of the study. And using some people who are much cleverer than myself, physiologists, pharmacologists and mathematicians all got together and came up with this idea of tissue reference modelling. So basically what this involves is a series of differential equations to model specific aspects of the transfer of radiotracer from the blood and between the unbound and bound compartment of that tissue model. I don't want to go into too much detail here because it is quite complex, but the issue is that it requires repeat arterial blood sampling and indeed metabolite correction because the parent radiotracer is constantly being metabolized in blood. And so this is exceptionally difficult. It's not impossible, however. There is a way around this, however. If you have an area within the body that is devoid of your target, you can use a reference region to basically model this blood component. But it's also difficult and more often than not impossible because it's very rare to find an area of the body that doesn't have your target issue. And then finally, one and the most popular way is to use an area as a reference that is unaffected by your disease or drug. And this is known as the pseudo-reference region, but it needs validating using a gold standard arterial blood sampling and metabolite correction to really pass muster. So of course it's very difficult. But we're here at ARA-MBI to try and help you get around these limitations in analyses. So as Rob mentioned, we have access to the Twilite 3, which is an inline blood radiation monitoring system. And what it does is we externalize the blood flow of the animal via an arteriovenous shunt, putting it through a radiation detector and then back into the animal. The nice thing about this is that it limits blood loss, and this has obviously issues for animal welfare, but also for quantification, because if you're constantly leaking blood for the analysis of input functions, then you're removing radio tracer from your system, and this can obviously create bias in your results. It's also exceptionally easy for blood sampling. You just pull a line, pull a draw with really no mess, no fuss. Now, most importantly, it really reduces people power. Now, back in the, as some would say, the good old days, these experiments would require, 3, 4, 5 people constantly running back and forth between the animal and the gamma counter, with, limited resolution for how specific these measurements were. And this Twilite blood sampler really gets around all of these. So, and we are currently the only group in Australia to have access to this. So as I said, we also have a radio HPLC and gamma counter that we can use for metabolite correction, and also to get around potential issues of binding of parent compound to plasma proteins within the blood, so plasma-free fraction. And then finally, we have some fantastic software, PMOD, which really enables plug-and-play quantification, so you don't have to be an absolute expert in kinetic modelling to apply this application. And of course, we can take you through the process here. And then finally, we also have some fancy kits that will help with biodistribution. So the gamma counter, but we also have the capacity to do autoradiography on thin sections, and this is just an example here to the right. So one approach is to do it ex vivo. So where we apply the radio tracer, and we can or cannot do a PET scan, and then we cull the animals afterwards and cut frozen sections on a dedicated radioactive cryostat. And the resolution is vastly improved. So in case you've got a region that, regions that are close by that might have differential effects, this really lets you hammer home what region is potentially involved in your disease state. The nice thing is that once these slides or sections decay, you can then go on and use these same sections for histology. So you can directly compare how much target there is in your area of interest to any other histological measure that you can think of. We also have the capacity for in vitro autoradiography, which is basically applying radio tracer directly to our frozen sections. And this is a really useful approach in the development of novel radio tracers. It's basically a first pass, so assessing whether or not my radio tracer binds to my target of interest. We also, using this approach, can really directly quantify binding affinity, so how well a radioligand binds to a receptor, and also receptor number. So you're getting down to these micro parameters I was talking about before. And then we can also, because it occurs in vitro, in the absence of any endogenous ligand, we can also assess whether or not endogenous ligands are having an effect or an influence on that PET signal. So altogether, a very nifty technique, and we have all the bits of kit that you need to be able to do these experiments. Okay, and so I'd just like to finish by acknowledging some of our users whose images I used today, as well as my wonderful radiochemistry team over at Peter McCallum, who also provide the radio tracers for those images in question. And then, don't send them to me for a CAT scan. Definitely send them to me for a PET scan. And if you have any questions, my email is there, and I think we're also going to stay on for a little bit. So thank you, everyone.
All right. We'll just have a quick flick through any questions that may have come through, and we'll also open up the floor to any additional questions. So First question is, have you already selected the cyclotron brand and are joining radiochemistry hot cells and synthesizers, or is this tender process still ongoing? So I think this is a good one for you, Maggie, clearly related to our cyclotron.
Sorry, the tender process is still ongoing. So we haven't made any decisions yet.
Right. This one is for Bianca. So what is your strategy to select a new PET tracer to test preclinically, assuming that the medical chemist can come up with greater than 5 different useful structures and you want to maximise your chances of success?
Yeah, great question, Giancarlo. Obviously, there's a number of different processes that we go through that, as you will know, there are different pharmacokinetic, binding characteristics, PK, just to name a few, that we tick off to determine which is our best structures to go forward with, but it's very much an iterative process and is fraught with difficulty. But yes, that's something that we go through in close collaboration with our radiochemists. So.
Great. And the last question that we've got is on the Twilite approach. Do you perform time corrections on the data, mostly the input function, for the extra time blood spent in the extra body routing?
Yeah, so again, another great question, Giancarlo. So this is obviously a potential issue, but the tubing that we use is exceptionally thin, and the line that we use to externalize is as short as possible to try and really limit the potential for delays in sampling to influence our binding assessment.
Great, wonderful. I think we may have had a new question come through. No, I think that's it. So if we don't have any other questions, we might end it there. So thank you to everyone for attending our showcase of our capabilities here at MBI and ARA-MBI. Please, if you've got any other questions or if you want to come for a tour of our facilities or anything, please don't hesitate to reach out. There's the 2 role accounts that oversee all of the facility inquiries, or feel free to reach out to Maggie or Bianca directly. So thank you all again for attending, and see you around. Thank you.